Here we see about the citric acid cycle. It is also known as Krebs cycle because Krebs invented the cycle. For for this, he got a Nobel Prize. And the cycle, also known as tricarboxylic acid, because three carboxylic acids are synthesized in the cycle. So there are fewer name we called upon this. So this is the central pathway of carbohydrate, lipid and amino acid metabolism. So it is the common pathway for all the metabolism. The citric acid cycle is a sequence of reaction occurred in mitochondria. So site of the citric acid cycle is the mitochondria because the enzymes are present inside of that. It is the final common pathway for the oxidation of these common carbohydrate, lipid, protein, because glucose, fatty acids, and most amino acids are metabolized into acetyl CoA. From the carbohydrate, the pyruvate or lactate converted into acetyl CoA. Here, the lipid are broken down into fatty acids. So, the fatty acids converted into acetyl CoA. From the protein, we get the amino acid during digestion, and this is also can convert into acetyl CoA. So this is the intermediates or precursor of the citric acid cycle and many of the products. The citric acid cycle also has a central role in gluconeogenesis also, which means the glucose synthesized from the non-carbohydrate sources. Next, the lipogenesis. Lipogenesis is the process in which lipid is synthesized from the carbohydrates, and the interconversion of amino acids also. So this is the important, very essential intermediate molecule produced after the metabolism. First, first we see about the startup reaction. The reactions of citric acid cycle, every step is carried out inside of mitochondria. First step, the acetyl CoA and oxaloacetate are condensed to form citrate, which means the acetyl CoA and oxaloacetate are condensed to form citrate, catalyzed by citrate synthase. Citrate is synthesized in this, so the name of enzyme is citrate synthase. In this step, a coenzyme molecule is released from the acetyl CoA. The next step, the citrate is isomerized to isocitrate by the enzyme aconitase, also citrate isomerase. So both are isomers. So citrate is just the positional changes occur inside of that. Then third step, isocitrate undergoes dehydrogenation, the decarboxylation to form a alpha ketoglutarate. The reaction is catalyzed by isocitrate dehydrogenase. So, dehydrogenase enzyme is involved. What is the process? Dehydrogenation. So, water molecules is released. And the next, the NAD is converted into NADPH. Next, the alpha ketoglutarate undergoes oxidative decarboxylation, and it is catalyzed by alpha ketoglutarate dehydrogenase complex forming succinyl CoA. So the final product of this reaction is succinyl CoA. The succinyl CoA is converted to succinate by the enzyme succinate thiokinase. So succinic acid, also known as succinic acid. Next, after that, the dehydrogenation of succinate forming fumarate. This is catalyzed by succinate dehydrogenase. So inside of that, a dehydrogenation process occur. Next, the fumarate is hydrated to L-malate by fumarase. So fumarate is converted into L-malate. Then malate is dehydrogenated by malate dehydrogenase to yield oxaloacetate. Then again, this pathway is repeated cyclic way. So this is also known as citric acid cycle. So After the energetics of this cycle is, as a result of oxidation catalyzed by the dehydrogenases of citric acid cycle, three molecules of NADH and one of FADH2 are produced for each molecule of acetyl CoA in one turn of cycle. These reducing equivalents are transferred to the respirate chain. A reoxidation of each NADH result formation of 2.5 ATP. So finally, in addition, 1 ATP is formed by substrate level phosphorylation by succinate thiokinase.